Hi everyone. In the previous lecture, I explained you what are platelets, how platelets are formed, and what is the structure of platelets. In this class, let's continue with the role of platelets. What is the function of platelets? Let me start with this terminology. What is hemostasis? Don't get confused with homeostasis. Homeostasis is different, hemostasis is different. What is homeostasis? Maintenance of constancy in the internal environment is homeostasis. Hemostasis is referring to stoppage of bleeding. Stasis is constancy, stoppage. Heme refers to blood. Bleeding is arrested by what mechanism? You can press and stop the bleeding. So you have an injury in the vessel wall, it starts bleeding, and you press over the vessel wall, it stops bleeding. So this external force application to stop bleeding, is it hemostasis? No. Hemostasis is the spontaneous stoppage of bleeding by some internal, natural, or physiological processes. Hemostasis is spontaneous arrest of bleeding by physiological processes. There are various mechanisms within the circulation or within the body that acts to stop the bleeding. So what are the causes for blood loss? What are the causes for injury to the vessel wall? Can the blood vessel be damaged without any external trauma or external forces? Yes, there can be a damage to the blood vessel even with external trauma or external forces on the blood vessel. A mere shear, that is the friction of the blood flowing with the endothelium, can cause rupture of the blood vessels if the friction is too much, if the shear rate is too much, if the flow is of very high velocity. A high velocity of flow can also damage the rupture the vessel wall. So what happens when the vessel wall is ruptured? Let us explain. This is the blood vessel wall. The vessel wall is having three layers: tunica intima, tunica media, and tunica adventitia. The innermost, close to the lumen, this is the lumen. Close to the lumen, the first layer is tunica intima, and this tunica intima is made up of endothelial cells. This is intima. Tunica intima is made up of endothelial cells. These are flat squamous epithelial cells. Squamous epithelial cells. They are resting over the basement membrane. So this is the basement membrane for the squamous epithelial cell or the endothelial cells. These are endothelial cells. And below this endothelial cell is a layer of connective tissue. So subendothelial connective tissue. This connective tissue is made up of cells. The fibroblasts cells are there, and also the collagen fibers. These are the fibroblasts, and some collagen fibers I will draw here. These are collagen fibers. And also there are von Willebrand factors. So this is von Willebrand factor that is present in the extracellular matrix of the connective tissue. So this connective tissue is made up of fibroblasts cells. So these are fibroblasts, and this is the von Willebrand factor present in the extracellular matrix of connective tissue, and also these collagen fibers are present in the extracellular matrix. Collagen fibers. Below this connective tissue is the next layer, the outer layer. So this outer layer is what is this outer layer? Intima. Next is media. This is the media. Tunica media. This tunica media is made up of smooth muscles. Smooth muscles. And the outermost layer is the again a thin connective tissue layer, tunica adventitia. So when there is any injury to this vessel wall, what happens? This endothelium is stripped off. This endothelium is stripped off, and there is a damage to this portion. So this endothelium is stripped off, and there is a damage to this portion. The blood which is flowing can escape out of the vessel through this damaged site. The blood which is usually flowing in the circulation can escape through this portion. So this is blood loss, blood loss. Now we need to understand various mechanisms to prevent this blood loss through this portion of damaged portion. So this is the damaged portion now. Through this damaged portion, there is bleeding, and various mechanisms to prevent blood loss is hemostasis. So the first step is vasoconstriction. The first step is vasoconstriction. The second step is platelet plug formation. The third step is formation of clot. And finally, fourth is clot retraction. So these are the various steps in hemostasis. So injury to the vessel wall will result in blood loss. To prevent blood loss, these are the various mechanisms that are acting. The first step is vasoconstriction. What do you mean by vasoconstriction? The smooth muscles which are in the injured site, these smooth muscles which are in the injured site, they are stimulated, and the surrounding smooth muscles, these are also stimulated. These all smooth muscles are stimulated by the trauma itself. So because of the activation of these smooth muscles, they contract. When the smooth muscles in the wall of the blood vessel, they contract, the lumen decreases. The blood flow through the lumen decreases. The blood flow through the small that blood vessel decreases. So when there is decreased blood flow, the blood loss is reduced. So the first step is vasoconstriction because of contraction of the smooth muscles. So what is the explanation for vasoconstriction? The smooth muscle contraction, it can be because of the direct trauma. Trauma is nothing but injury. Injury or trauma to the smooth muscles can cause distortion or stretch of the smooth muscles. So this is like the mechanical stimulus to the smooth muscles. When there is some mechanical stimulus to the smooth muscles respond by contraction, resulting in vasoconstriction, decreased blood flow, and prevention of blood loss. Also, there are some mediators that are released. So these mediators, they are released from the damaged endothelial cell. See, this endothelial cell is damaged now. This damaged endothelial cell can release some mediator. This mediator can act on these smooth muscles. This can be endothelin. Endothelin is a vasoconstrictor that is released from the damaged endothelial cells, acts on the smooth muscles to cause vasocontraction or vasoconstriction. And also the platelets which are in circulation. See, the platelets were in circulation, right? In the circulation, there were platelets. They were in inactive form. So because of the damage, now what happens? These platelets which are in circulation, they can bind to the collagen which is present here. Exposed collagen. They can bind to the exposed collagen, and these platelets binding to the collagen fibers gets activated. This platelet gets activated. So what is present within the granule of the platelet? Serotonin is there. Thromboxane A2 is there. So serotonin, thromboxane A2 is also released from the platelets, though it is not present in the granules. The thromboxane A2 is produced by the platelet phospholipids turning into arachidonic acid. From the arachidonic acid, thromboxane A2 is formed, and these two, serotonin and thromboxane A2, that are secreted from these platelets, this will act on the smooth muscles. This will act on the smooth muscles, and the smooth muscles contract, resulting in vasoconstriction. So what is the explanation for vasoconstriction? The first explanation is the trauma directly acting on the smooth muscles, stimulating the smooth muscles like a mechanical stimulus, will result in spasm. Second is the role of vasoconstrictors. Endothelin released from the endothelial cells, serotonin and thromboxane A2 released from the activated platelets. The third explanation is, see, these smooth muscles are also innervated, right? The smooth muscles are also innervated. There is a nerve supply. Consider this is a nerve and nerve supplying to the smooth muscles. See, when there is damage, even this nerve is also damaged, or even this nerve is also getting activated. So there is some mechanical stimulus to this nerve, and this stimulated nerve can stimulate the smooth muscles to contract. So the muscles will contract because of the stimulus it is receiving from the nerves. So it is a neural reflex. The third mechanism is neural reflex or nerve reflex, resulting in vasoconstriction. So to summarize, the first step in hemostasis is vasoconstriction. Vasoconstriction is nothing but the contraction of the smooth muscles lining the wall of the blood vessels will result in narrowing of the lumen. Thereby, the blood flowing through the vessel wall is reduced and the blood loss is prevented. So, what are the factors that causes vasoconstriction are: one, local factor, that is the direct trauma acting as a mechanical stimulus for smooth muscles. The second is some chemical substances released either by the damaged endothelial cells, that is endothelin, or activated platelets. Serotonin or thromboxane A2 acts as vasoconstrictors acting on the smooth muscles for vasoconstriction. The third factor is the nerve which is getting stimulated during the trauma or injury can stimulate the smooth muscles to cause contraction of the smooth muscle and vasoconstriction. Next, platelet plug formation. How the role of platelets in plugging the injured site. So this is the site where there is damage of the vessel wall has happened and the blood is flowing. Now this site has to be plugged by accumulation and aggregation of more and more platelets here. More and more platelets have to get accumulated to form a plug. Let me explain how this plug is formed. This is the second step. The second step is formation of platelet plug. Let me explain how platelet plug is formed. So this is a blood vessel. The endothelium is stripped off. These are the endothelial cells. In this area, the endothelium is stripped off, exposing the subendothelial connective tissue. The collagen fibers are exposed, and also there are von Willebrand factors present in the extracellular matrix of the connective tissue. So usually, generally, the platelets, these platelets are in circulation, they are kept inactivated. These platelets will not have the property of adhesion, sticking to each other, or these platelets will not stick to the wall of the blood vessels. They are generally in circulation, they are inactive state. Only when there is damage, only when there is injury to the vessel wall, these platelets can stick to the vessel wall. So these platelets can stick to the wall of the vessels only at the site of injury. So these platelets now will come and bind to the exposed collagen and von Willebrand factor. The platelets will come and bind to the exposed collagen and von Willebrand factor, and in the last lecture I have explained you what are the different receptors through which these platelets will stick to the von Willebrand factor and collagen. So there are glycoprotein receptors present over the platelet membrane. So that is GP1B. This is the receptor present over the membrane, and this receptor attaches to the von Willebrand factor. So this is the von Willebrand factor attaching to the GP1B receptor on the platelet membrane, and there are receptors for binding to the collagen. So collagen will bind to these receptors present on the membrane of the platelets, and these platelets will get activated now. So these platelets will get activated when they bind to the von Willebrand factor. This is von Willebrand factor or collagen. Binding of platelets to von Willebrand factor and collagen resulting in activation of these bound platelets. The binding we call it as adhesion, and adhered platelets will get activated. Adhesion is because of binding of platelets to von Willebrand factor and collagen. So these adhered platelets now will get activated. What does it mean, activation? So there are some glycoprotein receptors present over the membrane. In the previous lecture I discussed it is glycoprotein 2B3A. These receptors are in inactive form when the platelets are in circulation. But when the platelets stick to this collagen or von Willebrand factor, these glycoprotein 2B3A gets activated. So what is the role of this glycoprotein receptor present on the platelet? They can stick to the fibrinogens. They can stick to the fibrinogens. So where are these fibrinogens from? They were in circulation along with the platelets. These fibrinogens were in circulation along with the platelets. So there are glycoprotein receptors that are getting activated. So now these fibrinogens stick to the glycoprotein receptors and they can link the platelets, neighboring platelets together as a bridge. This fibrinogen is a bridge between the two platelets binding to glycoprotein 2B3A receptors on neighboring platelets. And also these activated platelets will start releasing the contents of the granules. The contents of the granules are released. What are these contents of the granules? They are mainly ADP, thromboxane A2. ADP and thromboxane A2 are released from these activated platelets. So what is the role of these thromboxane A2 and ADP? These thromboxane A2 and ADP can bind to the receptors over the platelets of these. So ADP can bind to the receptor over the platelet. Thromboxane A2 can bind to the receptor over the platelets and cause activation of these platelets which are in circulation. So these activated platelets now will have glycoprotein 2B3A receptors which are inactive normally will get activated. So this platelet activated will have activated glycoprotein 2B3A receptors. And fibrinogen can stick to these glycoprotein 2B3A receptors. So there are receptors that is activated and fibrinogen can stick to these. So what happens? This will result in more and more platelet aggregation. So fibrinogen sticking to the neighboring platelets, activated platelets, and all these will get aggregated now. So this aggregated platelets mass will bind to these platelets. That results in formation of a plug. We call this as platelet plug. And covering the injured site. This hole is plugged by the platelets accumulated here. So these are aggregation of platelets. So the three steps in formation of platelet plug is platelet adhesion, that is the adhesion of the platelets which are exposed to the subendothelial collagen and von Willebrand factor. And this von Willebrand factor is a protein that sticks to the platelet, and this platelet will sticking to the wall of the vessels very strongly, even though any force in the blood circulation cannot detach the platelet which is sticking to the wall of the blood vessel. So this acts as a glue. Von Willebrand factor acts as a glue to make the platelet strongly bind to the blood vessel wall, which is denuded. We call it as denuded intima. Denuded tunica intima. So the platelets, these platelets will bind to the tunica intima which is stripped off. There is no endothelial cells. The endothelial cells are stripped off so that the subendothelial connective tissue is exposed. For this subendothelial connective tissue, the platelets come and stick. And von Willebrand factor is acting like a bridge. Von Willebrand factor is acting like a bridge to stick these platelets firmly to the wall of the damaged vessel. Once these platelets are sticking, that is adhesion of platelets to the wall of the blood vessels, these get activated. They start releasing the contents of the granules, mainly ADP and thromboxane A2. These two can activate the neighboring platelets so that more and more platelets express their glycoprotein 2B3A. This glycoprotein 2B3A, which was usually present over the membrane, and it was in inactive state. This glycoprotein 2B3A were inactive state. Only when ADP or thromboxane A2 acts on these platelets through their specific receptors, there are some chemical changes happening within the platelet, and this sends the signal to activate the glycoprotein 2b3a receptors, and this activated glycoprotein 2b3a receptors can bind to the fibrinogen. So they can bind to the fibrinogen, and binding of fibrinogen is such that the neighboring platelets are aggregated. The neighboring platelets are aggregated. So this fibrinogen is like a link between the neighboring platelets. So these platelets gets aggregated now to form a plug that covers the injured site and block the injured site so that the blood loss is arrested. If at all the injury is very small, the platelet plug itself is sufficient to block that site. Even in absence of the clot formation, a small hole can be plugged by the platelet plug itself and prevent the blood loss. So to summarize the role of platelets, the platelets in circulation, they are in inactive form. The platelets will get activated only when they bind to the injured site. So the first activator is the platelet binding to collagen, platelet binding to von Willebrand factor. So this activated platelet will start releasing the contents ADP, thromboxane A2. These ADP and thromboxane A2 will act on the neighboring platelets to activate these platelets. So all these platelets which were in circulation are getting activated. What does this mean? Activation means either they release their contents or the membrane glycoprotein which was in inactive state will get activated. So activation of glycoprotein 2b3a. So when this glycoprotein 2b3a is getting activated, it can now bind to the fibrinogen. So this fibrinogen binds to the glycoprotein 2b3a of neighboring platelets, linking the platelets to each other, resulting in aggregation of platelets. So if there is any receptor blocker for glycoprotein 2b3a, so GP 2b3a, if this is not getting activated, the fibrinogens cannot link the neighboring platelets and platelet aggregation is inhibited. So there are some drugs that can block these receptors. There are some drugs to block these receptors to prevent platelet aggregation. So these are abciximab, and see this thromboxane A2 that is released from the activated platelet will activate the neighboring platelets. This thromboxane A2 is produced from the cell membrane phospholipids, the lipid bilayer phospholipids. Cell membrane phospholipids will break down to form arachidonic acid, and from arachidonic acid the thromboxane A2 is formed, and one of the enzyme required for formation of thromboxane A2 is the cyclooxygenase COX. So any drug that can inhibit the cyclooxygenase will decrease the production of thromboxane A2, and if there is deficiency of thromboxane A2, the platelet activation and aggregation is suppressed. So the role of aspirin, you might have heard of aspirin. So this aspirin will inhibit this enzyme, cyclooxygenase enzyme is inhibited. Therefore, the thromboxane A2 production is suppressed, the levels of thromboxane A2 is reduced, and the platelet activation and aggregation is inhibited. So this aspirin is antiplatelet drug that reduces the thromboxane A2 levels, and the thromboxane A2 is supposed to be a platelet activator, and deficiency of this platelet activator will result in decreased platelet plug formation, antiplatelet role of aspirin. Also this ADP, what is the role of ADP here? ADP is also binding to the ADP receptors. ADP binding to the ADP receptors, in the previous lecture I explained you, those are purinergic receptors, and there are some drugs which will block these ADP receptors. They are drugs called clopidogrel. This clopidogrel is a drug that blocks the ADP receptors so that the ADP cannot bind and act on the platelets to cause activation and aggregation of platelets. So these drugs will suppress platelet activation, aggregation and formation of platelet plug. These are antiplatelet drugs. One is aspirin, clopidogrel and also abciximab. In the next lecture, I'll continue with the third step in hemostasis, that is how fibrin clot is formed, the coagulation factors role, and also the role of platelet in coagulation factors activation, the role of platelets in formation of clot along with the coagulation factors in the next lecture.